Hello everybody, and welcome to the first installment of a four-part series on the immune system and its functions. This web series was done as an honors contract for Illinois State University, and I figured it could also be helpful for anyone who is either a med student like myself, someone who's just taking a molecular and cellular biology class, such as BSC 197 here at State, or even someone who is just simply interested in the immune system and would be able to utilize this as an educational tool. Now, what we're going to go over in this series is mainly a recap and further explanation of everything you find in the immune system chapter of Campbell Biology, the 10th edition, which is chapter 43, like I have up here. And so anyone who is taking BSC 197 or is studying from Campbell Biology, I know some of you in the class with me, I told you guys about this. So basically what I'm going to do is I'm going to recap the chapter in detail and go over it so that it's not so strenuous to read it and go through everything. And this will be more of a visual approach to the chapter. So if you want to follow along, if you have the book, it begins on page 496. It's in the 7th unit, and it's chapter 43. It might be helpful to follow along if you can, because I'll be using a lot of figures from the book. I mean, I'll have them up here, but if you want to follow along with everything else while I talk, that's fine too. In order to explain things, I'll be defining a lot of vocabulary that the book uses, so everything that I have in here is from the book, all of the information, all of the figures, like everything. So in each of the 15 to 20 minute installments of this series, I'm going to go over one of the 4 parts in chapter 43. So this first video is going to be an introduction and into what's going on, and also part 1 of the chapter, which is about innate immunity in animals. As you right here, innate immunity. The second part is going to be about adaptive immunity and pathogen-specific recognition. After that, we're going to be talking about cellular defense against infection with bodily fluids and bodily cells. And in the 4th video, I will be talking about disruptions in the immune system. So, alright, let's just get into it. Let's get started. So, this introduction basically lays the groundwork and tells you the basic things that will progress into the first part in the book. So initially, it talks about pathogens and defines pathogens as any disease-causing agent, such as a bacterium or a fungus, a virus, or anything of that nature. It discusses that pathogens love internal environments of animals because it's a good living situation for them, which is sort of a parasitic relationship in the fact that hosts like us suffer in order for them to be able to thrive. Humans, because they're large, don't suffer as much as other organisms could, but anyway, the book describes the immune system as simply an adaptation that animals and plants have, that they have developed in order to fight this parasitic relationship. Now, the book defines the immune system as a culmination of a body's defenses, which enable the animal to avoid or limit infections. And it's also noteworthy that it says something doesn't have to necessarily be a pathogen in order to elicit a response, but many foreign objects will do that as well. So we won't get much into that, but it's suitable to recognize that foreign objects of all kinds elicit responses from the organism. Now, in the immune system, cells and fluids interact with each other and with pathogens in order to destroy the pathogens. So this figure right here is a good example, figure 43.1, in which a macrophage, which is the large brown object, is engulfing green bacteria. Now, it doesn't define what the macrophage is immediately, but a macrophage is simply a large molecule that engulfs other materials in order to absorb them. So a good example of a macrophage that will come up again later in the chapter is repeatedly is a white blood cell. So in organisms like humans, the immune system's first line of defense isn't internal things like the macrophages, but it's called barrier defense. So in humans, that's the skin and membranes that secrete out unwanted fluids. These include the lining of our digestive, respiratory, and reproductive tracts. And if a pathogen breaches a barrier defense, the immune system's next job is to distinguish if something is part of the system or not. So part of the organism. And the book describes that as distinguishing self from non-self. So how does the immune system do that? Well, that's what most of the chapter in the textbook goes over and explains, but the most simplistic answer right now is that the immune cells produce molecules called receptors that bind to foreign objects in order to activate defense responses. Now, this is called molecular recognition, and in the immune system, there's two specific types of molecular recognition. The first is innate, which we'll talk about in this video, and the second is called adaptive, which will be in the next couple videos. Now, figure 43.2, let me get to that. Figure 43.2 is right here. It's really good at distinguishing the differences between innate immunity and adaptive immunity. Innate immunity utilizes your barrier defenses. Now, like your skin, your mucous membranes, and is present in all animals and plants. The barrier defenses have a rapid response, and the internal component of them is encompassed by things such as killer cells and inflammatory responses. Now, adaptive immunity is separate in a couple of ways. First of all, it's not present in plants or invertebrates. Secondly, adaptive immunity is slower because it needs innate immunity to take place before it can be set into motion. Now, adaptive immunity is what encompasses the use of antibodies and cytotoxic cells, which both defend against infections. Now, continuing on with differences between the two that aren't necessarily in the chart, innate immunity utilizes a small set of receptors, while the adaptive immunity utilizes a large array of receptors, and therefore it requires more specificity in order to defend against pathogens. Now, adaptive immunity is activated after innate immunity has either responded to a pathogen or has failed to keep the pathogen out. Now, sometimes it's noteworthy to know that adaptive immunity, which can also be called acquired immunity, that's not what's noteworthy, is called adaptive because it compiles receptors as a response to previous exposures to pathogens. And so it's an adaptive feature of your immune system. It's something that changes over time and utilizes things that have happened so that it can become better. Now, moving on to features of innate immunity, which is what this section is really about. It's found in all animals and plants, and the book begins to talk about invertebrates especially because innate immunity is the only type of immunity that invertebrates have. They don't have adaptive immunity. Now, the book uses insects as an example of an organism that benefits greatly from innate immunity because insects have an exoskeleton composed mostly out of chitin, which is a very effective barrier against pathogens. Chitin also protects against pathogens that are ingested by the insect because it also lines their intestinal tract. So the book starts to depict lysosomes and hemocytes as the defenses for insects, And lysosomes are just enzymes that break down bacterial walls, and hemocytes are immune cells present in an insect's circulatory fluid. Now, in regards to mechanisms of innate immunity, figure 43.3, it's right here, this figure right here, shows a great description of the process of phagocytosis. So, phagocytosis is extremely important in cell immunity because a macrophage can simply engulf a pathogen in order to protect the other cells in the organism. In this figure, we can see a step-by-step process of how the phagocytic cell goes about engulfing the pathogen. So first, the pseudopodia, which just means false feet, surrounds the pathogens, which are purple in this figure. The pseudopodia then encompass the pathogen, and the pathogen is now engulfed by endocytosis. So after that, vacuoles within the macrophage form from the pseudopodia, encapsulating the pathogens. The vacuole then combines with a lysosome, and the compounds within the lysosome destroy the pathogen, and the debris from that pathogen is then released, unable to be an infection to the rest of the organism. Figure 43.4, which I believe is at the bottom of this page, shows a visual example of the innate immunity response in an insect by showing two flies, both of which have been engineered with GFP, which gives a fluorescent green glow upon the activation of an immune response. The fly on top was injected with, they both have GFP, but the fly on top was injected with bacteria in order to show the immune system response across the whole organism. So how do the cells in the immune system know what to do next and how to spread throughout the organism? Well, the immune cells bind, sorry about that. The immune cells bind to molecules found only on the outer walls of the pathogens. So in fungal cells, the cell walls contain polysaccharides, and bacterial cell walls have polymers of sugars and amino acids not found in animals. So the immune cells secrete proteins to recognize the specific characteristics of the pathogen to determine whether it's in the class of bacteria or fungi. So the book discusses how, because organisms secrete many distinct antimicrobial peptides in response to a single infection, it's hard to study the activity of a single peptide. But a solution to this is shown in figure 43.5, which is on the next page, I believe. So, in figure 43.5, which is right here, it's an inquiry figure, a scientist by the name of Bruno Lemaitre and his research team used genetic techniques to reprogram a fly's immune system and found that the synthesis of a single peptide could provide an effective and specific immune defense. So some background of the chart in the study is that the research team engineered the flies to express significant amounts of a single peptide and measured over time whether or not that single peptide could keep the flies alive from an infection of a fungi or a bacteria. So the variable mutant in the graphs represents flies in which pathogens are recognized, but the signaling that would normally trigger an innate response is no longer functional. So the top chart represents the survival against the fungi, and the bottom chart represents the survival against the bacteria. And actually, you guys probably can't see that. So up here is just the background of the experiment, but down here are the actual charts. So this is the chart. On the top is survival against the fungi, and bottom is against the bacteria. So the chart shows that the wild type did relatively well at keeping the flies alive, while the fungi and bacteria, and the mutant plus drosymicin, maybe? I don't know how to say that properly, but it was an effective defense against the fungi, but not the bacteria. So the mutant with defensin worked well against the bacteria, but not against the fungi, and the mutant alone did not prevent well against either pathogen. So, progressing from invertebrates to vertebrates, we also progress from talking only about innate immunity to talking about a combination of innate and adaptive immunity again. So, the book focuses on mammals as a point of study for vertebrates, and their reasoning is that most studies regarding vertebrate immunity have come from both mice and humans, and the application here is, especially for people like me who are med students, the application we want here is for humans anyway. So, as we had talked about before, the first line of defense in an organism's innate immunity are the barrier defenses. So, the fluids that mucous membranes secrete not only physically remove pathogens from the organism, but the environment that the fluids possess are hostile to the pathogens. Tears, saliva, and mucus secretions have lysosomes that destroy cell walls of bacteria as they enter openings in any sort of tract in the organism. Stomach acid is another fluid that is effective at removing pathogens because when the pathogen is ingested, they have to contend with the acidic environment. So similarly, oils and sweat glands give human skin a pH ranging from 3 to 5, which is acidic enough to prevent bacteria from reproducing effectively. So, pathogens that defeat barrier defenses are then taken in by phagocytic cells. These cells use several types of receptors to detect different types of pathogens. Now, mammalian receptors are very similar to the toll receptors of insects, and the book defines a mammalian toll-like receptor, which is abbreviated TLR. If it would scroll up, oh, there it goes, scrolled up too much. It describes this as a membrane receptor on a phagocytic white blood cell that recognizes fragments of molecules common to a set of pathogens. So figure 43.6, which is down here a little ways, it's right here, shows the different TLRs. Now TLR3, which is on the inner surface of vesicles formed by endocytosis, binds to double-stranded RNA. TLR4, located on the plasma membrane, recognizes lipopolysaccharides, which is found on the surface of many bacteria. TLR5 recognizes flagellin, which is the main protein of bacterial flagella. And TLR9, which can be seen inside the vesicle with TLR3, is binding to a sequence of DNA rich in cytosine and guanine. Next, we will be talking about the separate types of phagocytes, which are neutrophils and macrophages mainly, but dendritic cells and eosinophils are also included in this. So neutrophils circulate in the blood and are attracted by signals from infected tissue. Macrophages, so this is neutrophils, macrophages are large phagocytes that sometimes migrate throughout the body. Others remain in organs or tissues and are where they are likely to encounter other pathogens. The other two types of phagocytes provide additional functions for innate defense. Dendritic cells reside in tissues like the skin that contact the environment. These types of cells stimulate adaptive immunity against pathogens that they encounter. And then finally, eosinophils are important for defending against multicellular invaders. You can see here that the example the book gives is parasitic worms. When the eosinophils encounter the pathogen, they excrete catabolic enzymes. Another type of immunity important to innate immunity, that is type of cell, that is not phagocytic is the natural killer cells. So these are white blood cells that can kill tumor cells and virus-infected cells as part of innate immunity. But instead of engulfing the cell like a phagocyte would, they release chemicals that lead to the death of the pathogen and inhibit the spread of the virus or the cancer. So to introduce a new concept, lots of the cellular innate defenses in vertebrates involve the lymphatic system. The lymphatic system is simply a network that distributes a colorless fluid called lymph throughout the body. Lymph is mostly made up of water and white blood cells, but lymph nodes, which are organ-like lobes throughout the body that contain the lymph fluid, are where some of the macrophages stay. So dendritic cells do not reside within the lymphatic system, but they migrate to lymph nodes after their interaction with pathogens. So after the arrival at the lymph nodes, dendritic cells interact with other cells to stimulate adaptive immunity. And then figure 43.7 gives us a great look at the lymphatic system. Shown in green are the lymphatic vessels through which the lymph fluid travels. Lymph nodes are colored in orange, and lymphoid organs such as the spleen, the tonsils, and the appendix are colored in yellow. The figure also shows the travel of lymph throughout the body in order to destroy pathogens. First, the fluid continually enters the lymphatic vessels, then the fluid flows through those vessels throughout the body. Once the fluid reaches a lymph node, pathogens and foreign particles that are present in the fluid encounter macrophages and are then destroyed. So the lymphatic vessels then return the fluid to the blood by two large ducts that drain from the veins, or from the shoulders into the veins. Alright, now we're going to move on from the lymphatic system to the types of antimicrobial peptides and proteins that are produced in the immune system. The book introduces interferons, which are proteins that produce innate defense by interfering with viral infections. Virus-infected cells secrete these interferons, which get nearby uninfected cells to produce substances that prevent the spread of the virus. This is how interferons limit cell-to-cell spread, helping to control viral infections such as colds and influenzas. Interferons can also sometimes help white blood cells enhance their phagocytic ability. Interferons are used in pharmaceuticals and are mass-produced to help treat viral infections. So from that on to inflammation. The inflammatory response is an innate immunity defense triggered by physical injury or infections of tissues, which release substances that promote swelling to enhance the concentration of white blood cells and tissue repair and the destruction of the invading pathogen. An important molecule that signals for inflammatory response is the histamine. Now, here's everything on interferons and inflammatory response. Histamines are substances released by mast cells that cause blood vessels to dilate and become more permeable in situations for inflammation and allergic responses. Now, when histamines cause blood vessels to dilate, the capillaries then leak fluid into neighboring tissues, which causes swelling. Macrophages and neutrophils modulate immune responses during inflammation, and cytokines are released by macrophages, and neutrophils promote blood flow to the infection. The increased local blood supply produces a redness and increase in temperature typical of an inflammatory response. So, during inflammation, there are cycles of signaling and response that transform their site. So, the figure at the bottom of the page, 43.8, shows the major events of an inflammatory response. At the site of an injury, what primarily happens is that the mast cells release histamines, which cause the capillary dilation, while macrophages release their cytokines. So in the first picture, you can see that the histamines and the histamines and the mast cells are both shown in red, while the macrophage and the cytokines are shown in blue. And in the second slide, you can see that the dilated capillaries are now allowing antimicrobial peptides to enter the tissue. And the accumulation of antimicrobial peptides coming into the tissue is what causes pus to form under skin. So pus is simply the fluid underneath the tissue that is rich in white blood cells and dead pathogens. And then in the final step here, we see that the neutrophils digest the pathogens and the cell debris, allowing the tissue to heal, and you can see that the damage is now reduced. Local infections can cause a small inflammatory response, but severe tissue damage can lead to a systematic response from the entire immune system. So cells and injured tissue can secrete molecules from bone marrow, and in the case of severe infections such as meningitis or appendicitis, white blood cells within the bloodstream may increase tenfold within only a few hours. Fever is an immune response that is considered inflammatory. When certain pathogens enter your body, substances are released by macrophages that cause the body's thermostat to reset to a higher temperature. This reset is good for fighting off certain infections, and it is hypothesized that the increased body temperature increases phagocytosis by accelerating tissue repair. So when certain bacterial infections are introduced, they can invoke the life-threatening condition that is known as septic shock from overwhelming systematic inflammatory response. This septic shock is characterized by very high fever, very low blood pressure, and poor blood flow. Septic shock is more susceptible to people who are very old or very young. Chronic inflammation is also an ongoing threat to humans' health. An example of this is Crohn's disease, which is often a debilitating condition in which an unregulated inflammatory response that is ongoing disrupts the intestinal function. One issue that the immune system must coordinate for things like this is that pathogens can adapt and evolve to be able to avoid destruction by phagocytic cells. One example of this is the bacterium Streptococcus pneumoniae, which can cause ear infections, meningitis, and pneumonia. So some bacteria, after being engulfed, can resist a breakdown. So in the first figure of the book, we had saw that large brown macrophage in the green bacteria. The green bacteria here are actually a bacteria known as Mycobacterium tuberculosis, and they are being engulfed by a white blood cell. But rather than being destroyed, these bacteria can sometimes grow and reproduce within this macrophage, using the macrophage as a sort of shell to defend against the body's immune system. And this ability to prevent destruction by the immune system can make certain bacteria and fungi substantial pathogenic threats to the world. So that is everything for chapter 43.1. I'm sorry it was so long. I'll try to cut down on the next couple. I didn't realize I had put too much information in this, and but in the next video we will be going over 43.2 and talking about pathogen-specific recognition and adaptive immunity. And I will see you then. Take care.